But to lead us up to that point, we're going to have a tour through the most neglected kingdom, but my favorite kingdom, with Emmy Bruns. Thank you.
Okay, is the mic on?
Yep.
You can hear me in the back? Okay, thank you. I'm delighted to be here, and I want to thank the organizers for inviting me. EEID, I've been coming for 10 years, and it's been just an academic home, so I'm thrilled to be here. And I'm extra thrilled to talk to you about, to kick off the persistence section and talk to you about plants, because plants are a phenomenal system for studying persistence, because plant diseases are everywhere. Plants are sort of awash in fungal parasites. They're like the stars. The more you look, the more you find. This would probably be better illustrated on the hike today. And start looking at the leaves and find all of the grim disease everywhere. And so plant diseases are everywhere, but of course, the world is green. And so it sets up this lovely question of how do plants persist in the face of pathogens? And so I'm going to walk through that. That's sort of the main theme today. How do plants persist? How do their fungal pathogens persist? And then hopefully I also tell you, what can we actually learn about persistence more broadly from studying plants? So I just want to jazz you up about plants too. And so I've been coming to EEID for 10 years, but I wanted to start by talking about something that happened 35 years ago, which is Janis Antonovics and his newly graduated student, Helen Alexander, began studying disease dynamics in a metapopulation of this little wildflower, unassuming wildflower, Silene latifolia, in southern Virginia along these roadsides. And they did this through a combination of population surveys and experimental plots. And I'll tell you a little bit about the fungus because it's pretty cool, so you can see why everybody got excited about it. So the fungus is called Microbotrium lignatus thioichi. That's the full Latin name, but at the time it used to be a Eustalago still. It causes a disease called anther smut disease. This is really a sort of fascinating fungal disease. The fungus invades the plant systemically, gets in, and it sporulates in the anthers of the plant. So this is the pollen-producing part of the plant. And this is pretty bad for the plant, can't produce pollen. It also sterilizes the ovary. So it's a sterilizing pathogen, doesn't cause any mortality. And the transmission mode is really beautiful because you can see that pollinators, when they visit, can pick up the spores and move them to healthy plants. And it's really a disease of perennial plants. And two other things that make this a really nice system. One is for ecology, you can see it, and that seems sort of trivial, but when you walk around, we can just take disease prevalence data. You can just count the disease in healthy plants. You can actually even do it, and this is what Janis was doing 35 years ago, by car. So you drive very slowly and count the plants as you go along. And also, it's a totally unimportant disease. This plant has no economic importance whatsoever, which is actually great because it means we can study disease in a natural system and really try and understand what are the dynamics of persistence that are going on in a system where we're not trying to manage and eradicate a disease. So I think I sort of drove three major lessons from persistence out of this early work that Janis and Helen and colleagues were doing up at Mountain Lake. And they did this work through a combination of sort of classic population census, where you actually go to nature and see what's happening, and experimental populations, where you collect genotypes from nature, put them into cages, and allow transmission to occur so you can really see what's happening. And sort of three lessons that I think are really important that they took from this is, one is pathogens. They found pathogens persist at the metapopulation level. So you can see from these figures here that basically there's a lot of extinction colonization dynamics going along, and pathogens might not persist within a local population, but at the metapopulation level, they can persist over time. They also found that rapid evolution of resistance can facilitate host persistence. So they found that there was a tremendous amount of genetic variation in resistance, and that this could be rapidly selected for, and was really important for basically allowing the plants to persist in the face of disease. And then they also found that evolution of host resistance itself can actually drive the extinction of the pathogen. So as hosts evolve higher levels of resistance, this can actually be part of the cause of local pathogen extinction. And I would just say that these lessons have been sort of broadly supported by lots of other lovely work in plant systems. And I would encourage you to look at some of Annalisa's work on the Plantago powdery mildew system in Finland, and some lovely work on rust and wild flax in Australia. And all of these systems really emphasize the point that metapopulation dynamics are really important, and that evolution plays this key role in this sort of evolutionary dynamic plays a key role in driving these patterns of persistence over time. So that was 10 years ago. I met Giannis and fell into the anther smut world, which I have not regretted. And at the time he had teamed up with Michael Hood, and they had also decided to switch systems from working on that white campion to this really pretty little pink flower. This is Dianthus Dianthus pavonius, the alpine carnation. And it gets its own microbotrium species. So it's another smut, different smut species, same clade, and the biology of the disease is pretty similar. This host, though, only occurs in the Italian Alps, which I feel like had something to do with Giannis's decision to work on it. But it's also a really great system because although the biology of the anther smut and the fungus, the transmission is very similar to the white latifolia, the ecology of the host and its life history are really quite different. And it allows us to ask new questions. It allowed us to ask some new questions about persistence. And one of the first questions that I got involved in with this was asking, well, when does host persistence fail? And can disease actually drive populations to extinction? So is this a general rule that hosts can always persist in the face of disease? Or can we actually find examples where the disease can really drive extinction? And I just want to highlight a few points why we think this is important. So there's a couple of host extinction risk factors that this particular anther smut system has. One is that it's a sterilizing disease, and it's actually completely sterilizing. So when a plant gets infected, all the flowers are sterilized and it doesn't recover. So it's year after year, it's just producing smutty flowers. And remember, it doesn't have any effect on mortality. So infected individuals do not die. They remain in the population and they continue to spread disease. And really importantly, this high alpine flower is very long-lived. And so you get a long-lived plant that's infected and it just continues to produce disease. This can really drive population, really decrease the birth rate of a population and drive it towards extinction. Secondly, it's a pollinator transmitted disease, right? So it's basically a vector transmitted disease. So we would expect the dynamics to be sort of frequency dependent. And we've also shown that it can persist even as host density declines. So we find disease even in very low populations. So it doesn't have like a threshold density that would eradicate the disease before the host. And finally, the other sort of ecological thing that we found is that it just has incredibly high prevalence. So the population that I'm going to talk about that we were studying, 43% of the population is infected with this incurable sterilizing disease. And I'll illustrate this with some of our population genetic data. So this is where we work. This is Giannis's new spot that he picked out for us, and it's in the Western Italian Alps. And this whole meadow, that's France, sort of up over the mountains there. And this whole meadow in the summer is just pink with these dianthus flowers. It's really beautiful. And of course, 43% of these plants are infected. And so over time, one of the things we've been doing is just monitoring the host population. We go out every couple of years, set up a transect, count all of the flowers and count how many are healthy and diseased. Because as I said before, you can just do this. And this is what we found so far, these really dramatic patterns where we find that the number of plants shown here, this is 2 replicate transects we've been looking at. So the number of plants shown here on the left axis in these dashed lines has been dramatically decreasing over time. But the proportion of disease, shown here on the right axis and in the black line, that's been sort of rock steady at 40% or even increasing in transect 2. So it really sort of looked to us like maybe this is a case where disease is really going to tank the host population. But of course, we can't predict something with just phenomenological data. So we also were good disease ecologists, and we went out and did a mark recapture study, which mark recapture studies are great to do in plants. We had a high recapture rate. And so we followed a bunch of flowering plants for 6 years. We'd go back with metal detectors, find them every year, record their status. And this gave us a bunch of great information. We learned sort of the mortality rate, the transition to flowering rates, transmission rates, infection rates, recovery rates. And we put this all into a model. We just used a basic SI model and parameterized it with this data that we'd collected. And I'm going to kind of skim through some of this. But basically what we found then is that we said, well, great, let's predict what we found in the field with our model, and we did a really bad job with this. So you can see the observed disease is that black line there, and then all of our predictions, which we were pulling from our distributions of our parameters, were consistently under-predicting disease. And basically, we had under-predicted transmission. So we just couldn't, the level of transmission that we found was so low, there was no way we could maintain the disease at the 40% that we observed. And what we found over time is basically that during this mark recapture study, we had only marked adult flowering plants, as we assumed all the transmission was occurring between butterflies and bees moving the pollinators, moving the spores around. But we've since found is that there's actually a second transmission route, and this is aerial transmission to seedlings. And we have a couple lines of evidence. I'm going to share two of them with you. One is that we found that seedlings through inoculation experiments, we just collected up a bunch of different inoculation experiments we'd done in the greenhouse. And anytime I inoculated plants at the seedling stage, they got infected at this really high rate, like 80% of them became infected. If you do the same thing with the same inoculum at the adult stage when they're already flowering, 3 to 4% get infected. So there's this incredible difference in susceptibility. And then we also have found that there is some local level of spore dispersal that just occurs when the spores fall off the plants. And we can determine this by putting spore traps out and counting the number of spores that fall around the plant. And it's quite localized, but this kind of these spores could then land on seedlings and then tap into this incredibly susceptible host resource. So we went back to our model and fixed it to add this second transmission route, where we had now a density dependent transmission route to seedlings that accounted for this higher level of susceptibility. And when we put all this into the model, so this is basically a mixed vector and aerial transmission model, we get a lot better at now we can sort of predict the amount of disease that we actually see in the field. And so what we found from this is that actually aerial transmission, when we analyze this model, aerial transmission turns out to be completely critical for disease persistence in the system. So our model estimates that actually 70 to 80% of the total transmission events in the system are due to seedlings, like aerial transmission to seedlings. And we've also found that the disease cannot persist without seedling transmission. So if you take the seedling transmission away, the disease goes extinct. And we've also found that basically our original hypothesis that the host population would go extinct is incorrect. So the host population, if the disease goes extinct, the host population recovers. But even if the disease persists, we predict it's going to decrease over time to about 20%. We think maybe we were sort of seeing the peak of an infection. And so basically we have a situation where both the host and the pathogen are persisting. So the lessons for persistence, I think, that we can take away from this dianthus study here are, one, host extinction is actually unlikely, despite all of the early warning signs that made it seem like that. And this age specificity of resistance is really important for facilitating persistence of both the host and pathogen. So this high level of adult resistance is allowing the host to persist. This is why the hosts have not gone extinct, because they're incredibly resistant at the adult stage. But this high level of juvenile susceptibility that the plants maintain is allowing the disease to persist. So the question that my lab has been really interested in now is, well, why are the seedlings so susceptible? And this is sort of an evolutionary puzzle, because we would expect that reductions in survival early in life prior to reproduction would have the highest fitness consequences. It's bad to get infected before you've had a chance to reproduce. And so we've just been interested in this question of why don't we see the evolution of juvenile resistance? And there's a couple of different hypotheses. The one I want to talk about today is, well, maybe juvenile resistance is just more costly than adult resistance. We know that juveniles have more limited resources. For plants, young seedlings are often running on stored energy for a little bit until they can make their first true leaves. So maybe they just don't have extra resources to throw towards defense. So we hypothesized that maybe strong trade-offs could constrain the evolution of juvenile resistance. And for this question, we went back to, we stepped away from the dianthus system and went back to a system that's a lot easier to work with and one that we could work with locally, which is the original Silene latifolia that Janis started working on those many years ago in Mountain Lake. And so what we've done here is to try and measure these costs of resistance at different ages. We've collected a panel of 45 full Sib Silene latifolia families. These came from the original meta population in Southern Virginia. And we did a bunch of resistance phenotyping in the greenhouse. So we inoculated each family at different stages and looked at their age-specific resistance. And one thing I'll tell you right away is that there's actually no correlation in resistance between juvenile and adult resistance. So you can see here's each one of these circles is an individual plant family, and here's how much infection they get at the juvenile stage, the seedling stage, and here's how much infection they have at the adult stage. And so they're really separate traits here. And this is we found this in 2 other anther smut systems as well. And then we took those same 45 families and planted them out into a common garden in, this time in scenic Beltsville, Maryland, which was a little bit not quite as nice working conditions, but it's okay in the spring. And we had 16 replicates of each full Sib family, and then we just quantified fitness in the absence of disease for 2 years. And so here's our hypothesis. We predicted basically what we're going to be looking at is a trade-off in resistance. And this is a trade-off because we have infection rate here along the x-axis. And so things that have a high infection rate are more susceptible, and the resistant ones that get a low infection rate are over here. And we predict basically that fecundity in the absence of disease should be lower for the resistant plants. And what we thought was if there's a stronger cost of resistance for juvenile plants, that slope should be steeper for the juvenile plants than for the adult plants. And so here's some of what we started to find. Basically, we found that there was a tremendous variation in the amount of flowers these plants produced, and resistant plants actually produced fewer flowers in year 2 than susceptible plants. And this is for both ages. So this is 3-week-old seedlings on the left and 4-month-old adults over here, which is pretty consistent with a cost of resistance. But of course, this is one measure of fitness. This is just flowering in the 2nd year. And then we also measured flowering in the 1st year and survival over 2 years. And so we integrated all those fitness components in using these Aster statistical models that sort of integrates these life history traits. Because we also found sort of non-significant but minor effects showing that there's a correlation with higher mortality rate also associated with resistance. And so when we integrate these together, we actually find strong evidence for cost of resistance. So that, again, here's proportion infected along the x-axis, and this is this Aster expected cumulative flower number. But one of the things that you can find is that resistance is actually similarly costly in both adults and seedlings. So the slopes are not significantly different. So resistance is costly at both stages, but similarly so. So the answer to our question is juvenile resistance more costly than adult resistance? No, they're sort of equally costly at all stages. So we can't really explain, at this point, we can't chalk up the high susceptibility of juveniles just because it has a higher cost. So we asked basically, well, that's sort of the cost side of it, or the trade-off side of it. What about selection? Is there actually strong selection? How beneficial is juvenile resistance really? And for this, I took a modeling approach. And so started off with a very simple model, just juveniles maturing into adults, and then both can get infected. And for this, I just kept all the transmission rates the same, so it's the same currency, density dependent. And then basically, I ran simulations with a couple different genotypes. So I had here's age along here. So we have a juvenile class and an adult class, and we can compare basically an individual genotypes that are fully susceptible to ones that have what we see in the field, which is where they're completely susceptible at the juvenile stage, and then they jump to having high resistance at the adult stage, against another genotype that's just resistant sort of as soon as it's born and throughout its lifespan. And we assumed a cost of resistance, and that this was slightly more costly than this one. And so what we find when we do this is basically that we get this really interesting result where adult resistance actually, we simulate this by putting this into a fully susceptible population at 40% disease, which is what we found in the field. And adult resistance and juvenile resistance both initially increase in frequency because it's better to be resistant than to be susceptible when there's this much disease. But over time, you can see that the juvenile onset resistance gene is lost from the population and the adult onset resistance gene is maintained. And so we end up with a population where we have a polymorphism for this same phenotype that we find in the field, which is that the adults are really resistant and the babies are susceptible. And this is basically occurring because of an ecological feedback. So you can see that as disease resistance evolves, the amount of disease in the population declines, right? We can't maintain 40% disease when the population isn't fully susceptible anymore. So it drops, in this case, to about 18% prevalence. And as disease resistance evolves, the disease prevalence decreases. And so as the disease prevalence decreases, the average age at which an individual is most likely to first encounter the disease increases. And so basically as adult resistance evolves, it's changing the ecological conditions so that it's not worth it for seedlings to pay any sort of cost of resistance, because they're more likely to just escape disease probabilistically. And this is done in a more complex way. Also, Ben Ashby has sort of expanded a lot of this work too, and I would point you to that as well. And I wanted to mention one final paper that this is work that my colleague Ben Ashby and his talented grad student Lydia Buckingham have done. And they've now basically asked, well, can we take this same framework and explore how the trade-off structure affects juvenile susceptibility? So In this case, they did a model that allowed resistance to evolve independently at both the juvenile and adult stages and used adaptive dynamics to try and see sort of what's the optimal level of resistance for both these stages according to a cost. And so they tried a bunch of different shapes of trade-offs and types of trade-offs and different currencies of trade-offs. So they asked, well, what if the trade-off is with mortality, maturation rate, or adult fecundity? And I want to point to one result in their paper, which I got excited by. And this is basically, they showed that if both forms of resistance have a similar cost, so if you have a similar cost, and that cost is in fecundity, so there's a trade-off here between resistance and fecundity. And you do this with a sterilizing disease, that as you have more host sterility caused by infection, so here's where anther smut is over here at 1, you almost always end up with this situation where adult resistance evolves to a higher level than juvenile resistance. So even if the costs are really similar, the expectation is that we would actually have higher adult resistance evolve. And through the same mechanism of ecological feedback, you can see that in fact, here's the total host density, here's the infected host, and you can see that portion is widening. So it's a change in prevalence. And so I just remind you that this is in fact exactly what we found from the field, which is that we have costs of resistance in both cases. They're costs that are largely driven by fecundity trade-offs, and they're very equivalent. And so we think kind of that this combination of costs of resistance and these ecological feedbacks could be a really powerful way of explaining this high level of juvenile susceptibility that we see in nature and that's driving these persistence dynamics. And so just summarize by sort of giving you a little cartoon here. So this is kind of what we think is happening in nature. We've got this population of adults and seedlings. A pathogen comes in, and maybe one hypothesis that initially, if everybody's susceptible, there's really strong, the pathogen goes crazy, and there's these efficient pollinators that just move the spores exactly to the hosts. And there's this less efficient sort of aerial transmission mode where you're just getting guys that are right next to you. But this generates really strong selection in the host to evolve adult resistance and knock off that transmission mode. And so as that path becomes less tenable because the adults are really resistant now, this is going to reduce the pathogen population size. And it reduces it enough that we think there's actually quite weak selection for juvenile resistance. And so you're actually in this trap where you're maintaining disease because the selection isn't strong enough to drive juvenile resistance to fixation. And one question that we're really interested sort of in the future is, well, how do we think about coevolution now? So should the host would the pathogen try and overcome adult resistance, or is it going to fight back and take the path of least resistance and just specialize on the seedling? So that's a question that we're really interested in now and have some cool preliminary data on. And so I'll sort of wrap up by saying basically, I started this talk by giving you 3 major lessons for persistence that I thought had been generated by this lovely early work on anther smut, that pathogens must persist at the metapopulation level. I think we can say that metapopulation dynamics can be really important, but they can also persist within these large continuous populations. So this dianthus system is really not a metapopulation, it's these large swaths of continuous populations. This idea that rapid evolution of resistance can facilitate host persistence, this is totally held up by our data. But I'd also add that basically resistance doesn't occur evenly across host life history. So we now can think about resistance not just as a single trait, but as this more complex thing that actually varies across a host life history. And that the maintenance of juvenile susceptibility can actually facilitate pathogen persistence. And this occurs through sort of fecundity costs and these ecological feedbacks. And finally, if you took nothing else, I hope you'll be convinced that plants can be a pretty good model system for studying disease, and they're also really cute. And so with that, I just want to thank everybody. I thank my collaborators, Janis Antanovics and Michael, who have been instrumental in all of the dianthus work, and then a bunch of folks in my lab. Sam Slowinski in particular and Ali Keto have done all of the cost of resistance work and just grown a ton of plants. So thanks. Time for questions?
Excellent. We got some time for questions in the middle.
Hi, that was a great talk. Thanks so much. So Pete Thrall and Jeremy Burden's framework, the spatial framework, you have a perfect system to do something really interesting, because there's a push now. The thing they're missing is the total dispersal kernel with the multiple pathways in their framework. But there's a big push now led by Holdra Rogers and others to look at total dispersal kernels for plants with multiple pathways. And I'm lucky enough to be involved with that work. but I've never seen a system where you've perfectly got both the plant and the pollinator, plant and the pathogen dynamics. So you said in passing you've done spore dispersal.
Yeah, and I was amiss
mapping.
in so we actually just had a paper out that was led by Lawrence Eureka, where he was looking at this spatially as well, and also can confirm that both these transmission modes are important when you consider sort of the spatial distribution of disease on the landscape.
But mapping the multiple dispersal pathways for both the plant and the pathogen into a closed system that's geographically range limited would be a beautiful test of some of this total dispersal kernel type research as well. So I'll send you Haldra's paper.
Okay.
But just because that's missing from Pete and Jeremy's framework, this idea of multiple pathways. And I've never seen a system that would be so beautiful to look at that. So very exciting.
Great, let's talk.
Yeah.
Hi, thank you very much. It was really interesting. I just have one question. So with this feedback loop that you mentioned, for that to work, do you need adults to reach a certain level in and relative abundance in the system so that feedback can work? And would you say, would that then depend on the initial conditions of?
Yeah, so I didn't show that any of that work, but yeah, for that feedback to work, adult resistance has to be fairly pretty strong resistance. If you have moderate resistance, then you can evolve actually sort of the lifelong juvenile resistance instead if it's moderate. Yeah.
Hi, great stuff. What really sort of fascinated me is the fact that there was no correlation between adult and juvenile resistance. Do you know anything about either the physiological mechanisms or the genetics of that?
Both good questions that we're sort of actively looking at right now. We don't exactly know. We've done, we've done a little bit of work. Sam and some really great undergrads of his have done some really nice work looking at variation among strains and among families to try and get at this sort of co-evolutionary. And we know that certain strains of the fungus are actually, so if you look across adult resistance, what we find is that everything is very sort of concordant, and individuals that are resistant to pretty much all of the strains, but there's a lot more specificity at the seedling stage. So we think we're probably looking at, my guess is that as adults plants mature, they're bringing on a lot more resistance is coming online. So you're developing not just our genes, but bringing in different forms of quantitative resistance too. And so it's just more complex. Yeah, but we don't know what we're interested in doing is trying to figure out how much overlap is there in the genes between.
Allison in the back.
Hello. So you said these plants are sterile after they're infected. Hello. Is there any difference in pollinator visitation when these plants are sterile? And that might affect the transmission across the landscape?
Yeah. So we have looked at pollinator visitation rates, and they are biased against the diseased plants, and they favor the healthy plants. There's also a sex bias. So there's two sexes here, and they favor the hermaphrodites over the females.
Excellent. Thank you.